Okay, so welcome back to the Science Shack on a very, very cold January morning, but for something I think will warm the cockles of your biological hearts, the importance and everything we've learnt so far about genomes. So this is towards the end now of the gene expression topic for A-level biology. Basically, we're going to discuss here what we've learnt since the sort of dawn of the genetic era, okay, in broad terms, and how we know, right, what's out there. Okay, why are we doing these things? Why are we sequencing genes? Why are we sequencing genomes of organisms for huge amounts of time and energy? And what have we learnt as a result? So this is the bit of the spec here, the idea that sequencing projects have produced this wide range of organisms that we now know the genome sequences of. Obviously, simple organisms is very easy, and more complicated organisms like yourself are very much more difficult. Okay, important points here like the identification of potential antigens. Obviously, we all know a lot more about that than we did this time last year, and why it's more difficult in complicated organisms like us than it is in simple organisms like bacteria. Okay. The idea of exactly how we do this, the actual mechanisms, the biotech stuff, are continuously updated, constantly getting more efficient, but you don't actually have to know for A-level biology the ins and outs of this, okay? So just on that point, okay, this is the kind of image you get out of what's called a DNA sequencing machine. Okay, we call it Sanger sequencing. It's a highly automated process these days, a lot more efficient, quick and easy than it used to be. This process, a brief overview, uses like a modified version of what's called the PCR reaction, the same reaction that we use to test for COVID. There's a video on how that works in the Biotech Basics series, but we add in some fluorescent so-called dideoxynucleotides which terminate the chains. Okay, and you can look into how that works via the Biotech Basics series on this channel, but of course you don't have to know for this bit how it works, just accept that it does, although that's not very satisfactory if you've got a particularly scientific mind in terms of just accepting that it does. Okay, right, so let's move on, accepting that we can now sequence genomes very rapidly for a wide range of organisms, and start thinking about what sort of organisms have been sequenced. Okay, what sort of genomes have been sequenced? Well, these are some of what we call the higher organisms, the eukaryotes that we've sequenced. So you've got nematodes, fruit flies, mosquitoes, pufferfish, and of course humans, the Human Genome Project that was completed in the early 2000s. Okay. Now, why these things? Well, nematodes are a model animal. They're a simple animal that's used for understanding everything from sort of embryology and development, all sorts of other experiments. Fruit flies, Drosophila, are a model animal. A lot of genetics experiments done in those. You might remember sex determination was first discovered with eye colour in Drosophila. Or a lot of neurobiology, a lot of the nervous wiring stuff, genes that control the nerves. In fruit flies, mosquitoes have been sequenced, of course, because they're a vector of malaria, which is the biggest killer of humans. And pufferfish here, look, is one that's been picked on because it's a vertebrate with a small genome. So obviously all these organisms down here have less in common with us than something like a pufferfish does, but lots of the vertebrates have got very, very, very large genomes. Okay, as you can see, look, in terms of genome sizes coming down here, this is what we're talking about. Now, the M stands for mega and the B stands for base pair. Okay, so mega base pair, 100 mega base pairs, 165 mega base pairs, and then up here we've got 3.2 giga base pairs. Okay, now obviously you see these units, kilo base pair, mega base pair, giga base pair, and they mean the same as they would for megabyte, whatever it is. So that the mega, giga, and kilo still mean the same things, but the B means base pairs. Okay, so there's a wide variety of genomes now sequenced. Obviously, I've nicked this table from the Wikipedia page, very poor research methodology on my front, of sequenced eukaryotes, and then compared that with the sequenced genomes page on prokaryotes. And there's a lot more prokaryotes that have been fully sequenced than eukaryotes. Okay, this is just the species of lactobacillus that have been sequenced. Okay, now from those numbers there, you can probably see why. Okay, from the numbers in the column on the side here, you can already see why. Look, so we've basically got just under 2 million bases or base pairs on their own. No megas, no gigas, nothing like that. Just sort of our in straight numbers. So of course these are a lot smaller in terms of their genome than any of the eukaryotes up here. And that's why they're much, much easier to sequence. It's not the only reason, but it's one of the major ones. Okay, so to give you a sort of straight comparison here, if you can look at some of these organisms here, so like a bacteriophage, viruses, they would only have 48 kilobase pairs. So obviously kilo is 1,000, just like 1,000 grams in a kilogram, 1,000 whales in a killer whale. Just checking you're paying attention. So 48 kilobase pairs of DNA in it, of nucleic acids. That codes for 60 genes. Okay, and that's all the virus needs. As we go up to E. coli, as you can see, about 100 times ish more complicated. Okay, and similarly, we've got about 100 times more genes. So there's a kind of a linear correlation there. We go up to our yeast here, okay, Saccharomyces cerevisiae, standard brewer's yeast, okay, and we've got 13.5 kilobase pairs, but only 6,000 genes. Okay, so the linear kind of increase kind of stops there. And you should be able to spot what the difference is here between bacteria and yeast and fruit flies and humans, is that we cross into eukaryotes. Okay, and if you've been paying attention throughout this series, you will remember all the stuff about non-coding DNA. One of the main reasons why we start seeing fewer genes per base, if you like, less genetic efficiency, is because of that non-coding DNA. Now, in these lower organisms, extra DNA would actually cause them a massive survival issue. Okay, whereas with us, it doesn't so much. So there's not as much a selection pressure to reduce the amount of bases and reproduce faster and faster and faster. Okay, so why are these projects important? So that's the kind of numbers that we're dealing with, and quantities of DNA and the sort of organisms that we've sequenced. But why are we doing this? Right? What's the importance? Well, here on the left, look, you can see a picture of the human X chromosome. Okay, now obviously you should know a lot about the X chromosome from the genetics topic. The fact that in humans, women have one, men have two. Sorry, the other way around. Women have two, men have one. It's the other way around in birds, weirdly. But there we go. And all of these genes here, the sum of the genes that code for problems, genetic conditions, that they're on the X chromosome, which means they affect men disproportionately to women. Okay, so we've got huge medical advances that have come from sequencing, right? We've found loads and loads and loads and loads of these variations in the chromosomes that seem to be relevant, that are linked to particular diseases, disorders of a genetic nature. Okay, 1.4 million, right, of these so-called single nucleotide polymorphisms in total have been found. Okay, and we'll get to what an SNP, a single nuclear polymorphism, is in a moment, but you can probably work it out from the name. Okay, so once we know all these things and we know which SNP, which single nucleotide polymorphisms are relevant to which diseases, it's quite easy then to look for those polymorphisms in embryos, fetuses, in parents, in all sorts of things, in people, in adults, and look for what it is that they might either have or pass on to their offspring. Okay, and there are really famous ones here, like haemophilia B and A is X-linked, colour blindness famously affects men more than women. But there's a whole bunch of other ones, so cleft palate, some deafness, all those kind of different genes on there. Okay, so that's why understanding what is in the genome is important. Okay, in terms of us. So this thing called a single nucleotide polymorphism is exactly what it sounds like. It's at a single nucleotide, you've got a different base there. And this pops up in more than 1% of the population. So it tends to be, if you're looking for one, you might get a lot of a group of people who are suffering with one particular genetic disorder, and then a group of control people who are not, and essentially sequence their genomes and look for differences. And if you've got a consistent difference in that group of people, in the group of people suffering from an illness and the group of people who are not, then you've got a pretty good idea that might well be something to do with the illness itself or the condition itself. I mean, a lot of these single nuclear polymorphisms will make no difference whatsoever. You've only got to look back at the year 12, the AS level transcription translation lessons to understand why, but a lot of them are disease relevant. Okay, so we only call it really a single nucleotide polymorphism if it's to some appreciable degree within a population, so greater than 1% will have it. And SNPs underlined, as you can see, there's a few examples there at the bottom. So you've got things like sickle cell anemia, beta thalassemia, and cystic fibrosis, all of which you should have come across probably at GCSE level, if not at A level. All of those result from single nucleotide polymorphisms. So that's basically what we can find out from these genome projects, is where these single nucleotide polymorphisms are and what they do. Right, so that's your eukaryotes. Let's flip over briefly to prokaryotes here. Okay, now with prokaryotes, sequencing is relatively easy because they've got very short genomes. They're not attached to proteins. They are in this one big circular chromosome and often a few plasmids around the side. Okay, so most prokaryotes have got one circular piece of DNA. It's not associated with histones, there's proteins, and they've got no non-coding DNA. So what you see is what you get. The sequence in the DNA is the sequence on the RNA, is the sequence in the proteins. Okay, there's no splicing, there's no introns, there's none of that. There's no DNA outside of the genes. Okay, where if you remember your year 12 stuff, only about 1 to 2% of our genome is actually coding for protein directly. The rest of it seems to have regulatory functions and be accidents of history and so on and so forth. Okay, so sequencing a prokaryote, dead easy. Sequencing us, less so. Okay, now a little recap on two key terms here, because obviously we're talking about the link between the DNA and the protein. So when we talk about the word genome, what we're saying is that's the complete set of all the genes in the cell, and proteome is all the proteins that the cell is able to produce. And as we've just said, with a prokaryote, it's dead easy. The genome gives us a very, very clear idea of what proteome is going to be like, okay? Whereas with eukaryotes, it's a lot more complicated, right? So that's why more prokaryotes have been sequenced, as well as their very small size, okay? So determining a simple organism's genome and proteome is quite easy. So let's have a look at some examples. So a bacteria that causes the flu, Haemophilus influenza, fully sequenced in 1995, 1,700 genes and 1.8 million bases. Okay, sequence that, lovely, job's a good one. Okay, loads of other ones have now been sequenced. Obviously, that's disease important. H. influenza is very important in terms of disease causing. Thousands of other prokaryotes and single-celled eukaryotes have been sequenced by this thing called the Human Microbiome Project, to basically look at all the non-human cells that live in and on you. And on that note, I'll make you a book recommendation, which I'm currently reading at the moment, which is this, I Contain Multitudes by Ed Yong. Basically, it's looking at our current understanding and our increasing understanding of the microbiome in and on us, and how we interact with it, and how it's important in everything from our digestion, obviously, to our immune function, to our mental health even, and growth and development and everything else. Okay, so that's one reason why it's important that we do this work, sequencing prokaryotes, because lots of them have a direct impact on our health, either for good or for bad. Okay, however, right, there are some other more obvious, more pressing reasons why it's a good idea to have a good understanding and genetic sequence of every virus, bacteria, fungi that might interact with us. Okay, so it's hoped that info gained from this can cure disease, provide knowledge of genes that can be exploited, and one really famous example at the moment, for example, those genes that code for antigens. Scientists can use in the lab theoretically and design sequences, right, model the proteins made and potentially use them as vaccines. Now, where do you think I'm going with this? Well, of course, the COVID vaccine has only been able to be, or vaccines have only been able to be produced so quickly, I mean like human first quickly, because of our understanding of Genome Projects. So let me give you an idea of that timeline, okay, for COVID-19. So it was only on the 31st of December in 2019 that the Wuhan Municipal Health Commission in Wuhan City, Hubei Province, China, reported a cluster of pneumonia cases with a link to a Wuhan Hanan seafood wholesale market. Okay, we all know that story now. It was only on the 9th of January that China's Centre for Disease Control reported this novel coronavirus that was later named SARS-CoV-2. And it was then on the 10th of January, a Friday, and you'll see why that's important in a moment, that the first novel coronavirus genome sequence was made publicly available. So a day after they reported this virus, got this virus, the next day, bosh, there's the genome. Okay, sequenced via GenBank database, so internationally scientists could get to work on this genome. That is incredible, and it would have been unheard of, a number of decades ago. Okay, before easy sequencing of genomes. Okay, so incredible work. And the reason I mentioned it's a Friday is because then we get on to, okay, we know the sequence, what did we do with it, right? So let me introduce you to Professor Sarah Gilbert, who was the absolute right expert in the right place at the right time. She is a professor of vaccinology at the University of Oxford and co-founder of Vaccitech. Right, so this is a quote about her work from her colleague here, Associate Professor Teresa Lamb, right, as she's there on the side. So this is her talking about Sarah Gilbert's contribution. She said, "We were quick," says her colleague at Oxford, Professor Teresa Lamb, "as Chinese scientists published the genetic virus of the new the genetic structure of the new virus. Over the weekend, the vaccine was pretty much designed." Okay, we went pretty fast with it. Now that is an understatement. So the AstraZeneca Oxford AstraZeneca vaccine that's going into people's arms was designed over a weekend, right? With on a laptop. And if you hear interviews with Professor Gilbert, says she basically did it at home on a laptop, got the sequence, designed the vaccine, started to manufacture it, and then you see the next thing here. Look, it took a few weeks to create a physical vaccine that worked against COVID in the lab. Then the first batch went into manufacture by early April. Unbelievable speed. Well done, these people, right? That's an absolutely sort of life-saving intervention that they made there using this genome technology. Okay, and of course you can now map huge quantities of human microbiome, all these different species and these different families. Obviously I can't go into the detail of this because it's such a detailed diagram, but you can find diagrams like this and read Ed Yong's book if you're interested in what kind of organisms live in and on you and our changing view towards them. Okay. Now, also, this genetic sequencing, even further, even more profound than that, in the 1970s, and again, you may know this story from GCSE, actually showed us that there was a whole branch of the tree of life that we'd completely missed. Okay, we'd split everything into prokaryote, eukaryote, so bacteria and all the small stuff over there, and eukaryote over here. But there was this group of bacteria called the methanogens, who produce methane. They find them in our gut, in the deep ocean, around black smokers. They're quite common, and tend to be in quite extreme environments. Turns out they're actually not prokaryotes and they're not eukaryotes. They're a whole other domain of life, the so-called archaea. And this is a guy here called Carl Voss, right? And Carl Voss identified these largely due to sequencing RNA sequences within different, what was then thought of just as prokaryotes, okay? And he came up with this whole new group of life, which turned out, yeah, absolutely, we've completely missed this for the entirety of scientific history, thanks to sequencing of nucleic acids and thanks to Genome Projects. Okay, so an incredible discovery, okay, that really has changed the way we look at the tree of life and entered the GCSE syllabus in the last update. Okay, so it's a lot more difficult, as we said, for determining a complex organism's proteome from the genome. Now, I've put this pie chart here up. This one is from the this one's from the year 12 version of this. So go back to that video and have a little look if you want to at the what all these different bits are, or the one on structure of DNA and chromosomes. For your purposes, we're going to skip straight into the idea that we have no non-coding DNA. You should know a bit about that already. Okay. Obviously, it's therefore more difficult to sequence which part of the DNA is responsible for the protein, because it's not so much looking for a needle in a haystack, it's looking for a needle in a haystack that's been cut up and has got bits of hay interspersed into the needle. Okay, so that's quite difficult. Okay, so we've also spent a lot of this topic talking about regulatory genes. Okay, there's genes that produce proteins that regulate other genes. Okay, so some code for proteins, some the RNA itself is functional. Think RNA interference, microRNAs, siRNAs, all these things, they have to be coded for by a bit of DNA. Okay, so that all wouldn't be directly coding for protein in the way that we learn about it in AS level biology. Okay, so it's a lot more complicated in you, but it's very important that we carry on with this work. Okay, now of course you will know about the Human Genome Project, officially conceived of in the mid-1980s, officially started in 1990, and its formal completion date of here it is, we are done, was the 14th of April 2003. I remember it well. I was in my first year at university, just coming up to my first year exams, when they published that. Obviously, that doesn't mean done, right? We've got this sequence. Well, what do we do with it now? So since then, right, in the last coming up to 20 years, a lot of sifting and research has been done. Tons of sifting, tons of looking through this wealth of information to find out what is going on. Okay, all these genes for disease and inheritance, all that kind of stuff. This was an incredible international collaboration, the Human Genome Project, publicly funded international collaboration, basically an example of good science, how it should be done. Okay, so what's the upshot now about 20 years later? Well, I had a little rummage on the NHS website, and this stuff just simply could not have existed, 20, maybe less, fewer than that, years ago. Okay, you can go to the page on the NHS, and obviously, it's recent because I've got that snip there, to get to a page about genetic and genomic testing, which will give you a bunch of information, broadly speaking, about what genetics and genomics is and what testing is. Okay, if you dig a bit deeper through the NHS, you can get some of their internal, so they're publicly available, but some of their more obscure sort of spreadsheets of the available genetic tests. And currently, there are 530 odd genes for unusual rare diseases that can be screened for via the NHS as of August 2020, which is when this document was published. Okay, and of course they have a genetic counselling service now, so they have people who are there to interpret and explain to you the clinical outcomes of any SNPs or any other mutations that are found in your genome. So it might be that you've got a family history of something, cancer, cystic fibrosis, or something more obscure than that, and you're considering having children, or you want to know what your future prognosis is, and you would go via this genetic testing and then into genetic counselling to achieve that. And that is a fantastic service that the NHS now can offer thanks to Genome Projects. And we're not too far away from having actual genetic therapies available as well, particularly for things like cystic fibrosis. Okay, so that's our thing on Genome Projects. We're largely going to leave it there. We have covered, I think you agree, those parts of the syllabus, why these things are so important. If you want to know more about the sequencing methods, do look at the Biotech Basics playlist. There's more on that. If you're following the A-level course through, then that is the next topic that we're going to go into, but we don't spend too much time on Sanger sequencing. So as a recap, here is what you should know. So we started with mutations, a recap and an addition to the AS level material. We then went into new material on gene regulation, transcription factors, post-transcriptional regulation, RNA interference, small interfering RNAs, cancer genes, their causes and their possible treatments, genome projects and their importance is what we've just finished. So if you're looking to get ahead now, go on to the Biotech Basics series. I'll put a link in the description. Thanks very much, and I'll see you next time. Cheers.